I've always been fascinated by how the thousands of chemical reactions that take place within living cells are controlled so that the right molecules are made in the right place and at the right time and at minimal efficiency in terms of the energy consumed. In the 1960s, it became clear that one of the ways in which this is done is by endowing enzymes with special properties. And nearly 50 years ago, I started to work on a new mechanism of enzyme regulation in which phosphate becomes attached or detached from proteins. And this is the attachment of phosphate is catalyzed by a class of enzymes called kinases and removed by another class of enzymes called phosphatases. So the attachment or removal of phosphate changes the shape of the protein and its ability to function in a great variety of ways. At the time I sort of got into this, it was thought to be a very specialized control mechanism confined to the regulation of carbohydrate metabolism. But over the sort of next 25 years or so, I was involved in identifying and characterizing many new kinases and phosphatases and in finding out how they were switched on and switched off by a variety of signals such as the hormone insulin, by growth factors, and by cell-damaging agents. So by the early 1990s, it had become clear from work in both my lab and many other labs around the world that this wasn't a specialized control mechanism after all, but it actually was involved in regulating almost all aspects of the life of living cells, including all of the events depicted on this slide and many more. When the human genome eventually was sequenced about 15 years ago, it became clear that there's over 500 kinases and 150 phosphatases. So several percent of all the proteins encoded by the human genome are actually these regulatory enzymes. We now know that most intracellular proteins contain at least some covalently bound phosphate, and that abnormalities in the amount of phosphate in proteins, called phosphorylation, is a cause of cancer and many other global diseases. But the idea that you could actually target these kinases and phosphatases to develop drugs to treat diseases was considered to be an impossible dream even in the mid-1990s. And that explains why no pharmaceutical industry had ever had the slightest interest in anything that I was doing. But the situation changed in 1995 when I had two pharmaceutical companies approached me, and this led to actually two wonderful collaborations in which we were able to establish that an anti-cancer drug and an anti-inflammatory drug were very potent and specific inhibitors of two of the enzymes that the kinases that we'd identified. So this was very exciting because it meant that it was going to become possible to develop drugs in this area. But from my standpoint, I was most interested in how these drugs would actually be wonderful reagents to help advance my own research. So I started to try and persuade a number of pharmaceutical companies to work with me to help develop many more of these molecules. Initially, I was very unsuccessful because most companies didn't believe that this was a feasible area at all. And the idea that you would have 6 companies working with an academic in order to make a lot of wonderful reagents for his own research, they weren't at all impressed. But eventually, I did manage to get this collaboration off the ground. the hardest bit being actually to persuade the lawyers of 6 pharmaceutical companies to sign a single agreement. So this collaboration, which we call the Division of Signal Transduction Therapy, or the DSTT for short, it started in 1998. It's still operating today. It's, I believe, Europe's largest and certainly its longest standing collaboration between academia and the pharmaceutical industry. Over this period, we've helped to launch and accelerate dozens of new kinase drug discovery programs, and probably its most important achievement is the development of dabrafenib, which is a new treatment for malignant skin cancer, which was developed by GlaxoSmithKline, which was approved for clinical use about 3 years ago. So, but in every venture like this, you need a lot of luck. And our biggest stroke of luck was that just after we started this collaboration, the first kinase inhibiting drug went into clinical trials. It was a molecule called Gleevec, which has actually transformed a fatal leukemia and an intestinal cancer called GIST, a gastrointestinal stromal tumor, into a manageable condition. It was the sort of the huge financial impact of this drug and especially its clinical impact that galvanized the whole pharmaceutical industry into action. Just to show you how impressive this molecule is, on the left-hand side you can see actually the intestine full of a huge gastrointestinal stromal tumor weighing several kilograms, but within 24 hours of going on this kinase inhibitor, it's completely disappeared, and you can see that the same patient 9 years later is still completely clean. So this actually shows a picture of the 2 people who were instrumental in getting this program through to clinical trials. Nick Lydon, who's actually a former student from my department at the University of Dundee many years ago, is the person who really initiated this program and drove it right through to clinical trials. And Brian Drucker is the clinician who demonstrated its amazing clinical efficiency. So where are we now in this area? So since Gleevec was approved, there's actually 30 other kinase drugs that have been approved for the treatment of cancer. Kinases account for something like half or more of all the cancer drug discovery programs in the pharmaceutical industry. You can see that they now make a large amount of money. And there's actually over 150 other drugs that target kinases that are currently undergoing clinical trials, of which about 25 are in the late stages of clinical trials. So it's pretty clear that we'll be approximately doubling the number of kinase drugs in the next 5 years or so. Here is the list of those. I've highlighted in red at the bottom the two where my lab has had some involvement, either through the discovery of the target in the case of MEK or in helping to launch the program in the case of dabrafenib. In the last 18 months, it's become clear that if you actually combine these 2 drugs, you actually get a much more effective drug that's actually much more slow to get drug resistance, and this was approved for clinical use about a year ago. So what's the future of kinase drug therapy? Remarkably, despite all of this action, less than 10% of the kinases encoded by the human genome have so far been targeted by pharmaceutical companies to develop drugs. So will drugs that target that switch off some of these other kinases also be useful in treating other major global diseases, particularly diseases other than cancer, where this area has not yet taken off? So I believe that this actually will be the case, and that the big testing ground will be to develop kinase drugs for the treatment of inflammatory and autoimmune diseases. So we're talking about diseases like arthritis, asthma, colitis, fibrosis, lupus, psoriasis, and sepsis. So about 8 years ago, I decided to completely stop everything else that was going on in my lab and to try and apply my trade to an area I knew nothing about, which was immunology. And it's been a great learning experience. I'm learning and still learning an enormous amount of immunology, but I believe that the technology that I've developed in other systems has a chance of making an impact there. And in the last sort of couple of years, I think we've got some very sort of promising data now that, for example, has shown that if you switch off kinases in the IRAC family, this is has a good chance of being able to prevent and perhaps treat lupus nephritis, one of a very serious autoimmune disease. We've also recently discovered that a particular subfamily of kinases that go under the acronym SIK convert a particular type of red blood cell from an anti-inflammatory to an inflammatory state. So, for example, if you this white blood cell called a macrophage is in an inflammatory or M1 state, this is the state you want it to be in order to fight invading pathogens like bacteria and viruses, if you, when this has done its job, you want to switch these macrophages to an anti-inflammatory state. Otherwise, you'll get very serious tissue damage, and this will lead to a lot of inflammatory diseases. And so we're sort of very excited that we, in collaboration with MRC Technology and also with my collaborator Nathaniel Gray at Harvard Medical School, we've been able to develop the first molecules which, by switching off these SIKs, can switch macrophages from the inflammatory to the anti-inflammatory state. Anyway, this is still early days, but I think it's sort of potentially, quite exciting. So let me sort of finish by just showing a picture of my building where I work. This is the School of Life Sciences at the University of Dundee, where there's now more than 1,000 scientists from over 60 countries. But I show this really just, of course, to remind me to thank the more than 50 or so PhD students and nearly 100 postdoctoral workers who've studied with me on these and many other problems over the past 44 years, and of course my many other colleagues and collaborators. And let me just sort of finish, though, by thanking the Academy for making me a corresponding member. It's a great honor, not of course just for me, but for all the people who work with me and for the institution in which I work. So thank you very much indeed.
Thank you, Sir Cohen. The question? Okay. Andrew?
Given the broad range of and prevalence of kinase and phosphatases, is there a hope for rational design of inhibitors, given one needs to hit the specificity of just one particular protein or one particular process?
Rational design, well, I think semi-rational design is already going on. Obviously, a lot of these programs in the past have started from random high-throughput screening of compound libraries, but once one has the three-dimensional structure of these molecules by X-ray crystallography and knows how the prototypic molecules bind, you can then actually use this information to more rationally design much more potent and specific inhibitors. Whether you'll ever need to completely eliminate screening procedures, who knows? It's always dangerous to say probably not, but I'll say probably not.
One last question.
So, Philip, my question is an extension, really, of that one. Given that the kinases are essential for every metabolic process, how specific are these drugs, and are there serious side effects?
Actually, the approved drugs are remarkably free of side effects compared to conventional chemotherapy. There are a few incredibly specific kinases that don't seem to touch a single other kinase in the human genome, but actually many of the approved ones do. They actually hit multiple kinases. This actually probably helps to some extent to avoid drug resistance, but in the case, for example, of Gleevec, it means you can actually treat 3 types of cancer which are caused by defects in 3 different kinases. So, for example, the molecule Gleevec I mentioned, it actually alleviates this form of leukemia called CML by switching off a kinase called ABL, but in the case of the gastrointestinal stromal tumor, it's doing this by switching off an entirely different kinase called c-KIT. So sometimes the lack of specificity can actually have some benefits. In the case of, I think in the case of cancer, you can get away with more than you can with other things like inflammatory and autoimmune diseases where, people may need to be on these drugs for 30, 40, or 50 years. But only time will tell, actually, whether, side effects will emerge after these many years of treatment.
Thank you, Sir.